Okay, we'll start the next session showcasing the talents of the DIM, starting with Benoit. Benoit Chassaing, voilà, tu es là. From Pasteur Institute, the floor is yours.
Okay, good morning everyone, and yeah, thank you for the invitation to present a little bit of what we are doing in the laboratory. And what I wanted to discuss today is really related to the project that was supported by the DIM, and how and why we think that mucus-associated bacteria are very important for health and disease. So very briefly to introduce and to give you an overview of what we are doing, we are really interested in the lab of the host-microbiota interaction at the mucosal surface, and especially how select bacteria are interacting with the mucus layer. So you can see here on the top of this picture, you have the bacteria that are labeled in red. Those little dots are bacteria that are labeled in red in the intestine. And then you have this very thick mucus layer in green that's lying on top of the epithelial mucosa that's labeled in purple. And this is at steady state, and we know that this is health associated, and this is a good situation. But what we are interested in the lab is trying to understand how we can switch to a disease-associated condition, which is presented here on the right picture, where you have now this bacteria able to penetrate and to colonize the inner mucus layer. You have full colonization of this mucus layer, and the bacteria getting in close contact with the epithelium. And we know that such microbiota encroachment within the mucus layer is always associated with chronic intestinal inflammation and disease condition. So we are working on multiple models in the lab. I just wanted to give you an overview of what we are doing on one of them, which is related to dietary emulsifiers. So very briefly, what are they? They are food additives that are highly used by the food industry to extend shelf life and improve texture of packaged products. And they are really used very, very broadly. And if you think about ice cream or everything that's packaged and processed, it will be loaded with dietary emulsifiers. And what we observed in the past is if you treat mice with dietary emulsifiers, this is going to be sufficient to alter the microbiota in a detrimental way. So you are going to alter the composition, and I'm really going pretty quick on this aspect because this was published quite some time ago now. You can alter the composition of the microbiota by giving those food additives to the mice. Not only do you alter the composition, so you alter which bacteria are present, but you also alter, very importantly, their interaction with the host. So this is what's represented here with the quantification of fecal flagellin. So flagellin is an antigen that's expressed by the microbiota, and that will be able to drive recognition by the innate and adaptive immune receptor, immune signaling, that will drive chronic inflammatory disease. And you can see that if you place the mice with two commonly used dietary emulsifiers, this microbiota is getting much more pro-inflammatory by expressing much higher level of fecal flagellin. And the second graph that's represented on the right part is regarding microbiota encroachment, so the concept I presented you before, where we are able to quantify the localization of the microbiota. And you can see that when you fed the mice with dietary emulsifiers, this microbiota is getting in close contact. So basically, if I move back to what I presented you before, you are going from the left picture where the microbiota is kept at a safe distance, and just by putting these food additives to the diet of the mice, you are going to the right picture where the microbiota now is getting much closer in a way that will drive chronic inflammation. So this is, of course, something that we investigated, for example, in mice model. And if you take, very briefly regarding the mechanism, you take mice, you give them dietary emulsifiers. This is sufficient to alter the microbiota in a way that will drive many types of diseases. So this is going to drive metabolic deregulation with increased fat deposition and increased adiposity, as well as type 2 diabetes in mice model. And on the right graph, those are mice genetically susceptible to develop colitis. And you can see that just by adding those food additives to the diet, the mice are indeed developing much more severe colitis compared to untreated animals. And regarding the mechanism, the microbiota is very key to drive this detrimental effect, because if you take germ-free mice, so mice without a microbiota, we are completely losing the phenotype. So dietary emulsifiers are not detrimental anymore. While if you take germ-free mice and on those mice you transplant the microbiota from emulsifier-treated animals, you can now fully recapitulate what we observed before. So clearly suggesting that the microbiota is really the direct target of those compounds in a way that will drive chronic inflammation. And we again have plenty of data showing that those mucus-associated bacteria are very key, are very central and key to drive chronic inflammation induced by those compounds. So of course this is bringing me to the human relevance, because everything I presented so far was using mice model. And regarding the human relevance, we have some data that was again published a couple of years ago in human with or without type 2 diabetes and metabolic deregulation. And what we observed is there is a perfect correlation between the severity of type 2 diabetes, which was quantified here by HbA1c level, and the severity of microbiota encroachment. So if you look at human with a very severe type 2 diabetes, they are having a mucus layer that's fully colonized by bacteria. While if you are looking for human without any metabolic deregulation, those mucus layers are fully intact and not colonized by microbiota. So of course this is a purely correlative approach, and of course the next very important step will be to identify and to test the causal link between those mucus-associated bacteria and metabolic deregulation that can be promoted by those mucus-associated bacteria. And if I'm going back to the dietary emulsifier concept, so there is also human data suggesting that those compounds can indeed be a very key player for chronic diseases in human. And this was, for example, reported by, thanks to the Nutrinet Santé cohort in France, showing that in patients that are in healthy individuals that are eating or drinking more dietary emulsifiers, they have an increased risk of various types of diseases, including type 2 diabetes and various types of cancer. This is, again, correlation analysis and epidemiological data, so it's not really telling you if dietary emulsifiers is sufficient per itself to drive the disease. So this is something that we tested in this pilot clinical trial that was, again, published, as you can see, 3 years ago, where we were able to feed dietary emulsifiers to healthy individuals and look at the effect on the microbiota and metabolic deregulation in those healthy participants. And what we observed is represented here, again, looking at mucus-associated bacteria, because we really think that this is very key in driving associated downstream chronic inflammatory condition. You can see in orange that for most of those participants in which we gave dietary emulsifiers, microbiota was not really altered in its localization and interaction with the mucus, while we observed that for 2 of those participants, they were characterized by a very strong microbiota encroachment following consumption of dietary emulsifiers. So it was kind of disappointing at the beginning, showing that there is this type of two group, and this was really bringing the concept that maybe we are not all equal in the human population, that we are not all equal when we will be facing those dietary emulsifiers, and that maybe some of us will be emulsifier resistant and some of us will be emulsifier sensitive. So this is something that we wanted to test, and for this, so as you can see, we went from mice to the human, and then after the human data, we went back to the mice because we wanted to understand a little bit more the mechanism. And what we did was to colonize a group of mice with the microbiota from either CMC sensitive or CMC resistant individuals. And what we observed after microbiota stabilization and emulsifier treatment, what we observed is in those 2 groups of mice that were colonized with a CMC resistant, so an emulsifier resistant microbiota, you can see that in those mice, if you treat them with emulsifiers, there is no inflammation anymore. Those mice are fully protected against inflammation normally induced by dietary emulsifiers. But if you do the same, but this time in mice colonized with CMC sensitive microbiota, you can see that now those mice are developing very strong colitis. So dietary emulsifiers are indeed sufficient to drive chronic inflammation, but for that, you need to host a very specific sensitive microbiota in order for the emulsifiers to have the downstream detrimental effects. So this is really leading me to the conclusion and to our working hypothesis, which is that we are not all equal when we'll be facing those compounds, those dietary emulsifiers. Maybe some of us are colonized by a resistant microbiota that will be fully protected and that will not respond to those compounds. And on the other side, you can have some microbiota that will be very sensitive, and we are still working on the mechanism in a way that when we will be facing those dietary emulsifiers, we will be responding, the microbiota will detect, will respond to those compounds in a way that will drive microbiota encroachment, so the mucus layer will be colonized in a way that will subsequently lead in the long term to chronic inflammatory condition and metabolic deregulation. So we really think that those mucus-associated bacteria are very key in driving those phenotype, and we are currently investigating and isolating those bacteria. And this is actually why we got the support of the DIM, was to acquire a cell sorter that is dedicated for bacteria, that allow us to detect and sort and cultivate, because everything is placed under an anaerobic condition, so we can keep the bacteria alive, to clearly collect very specifically those mucus-associated bacteria, isolate them, identify them, and then we can put them back to mice to see if by themselves they are indeed sufficient to drive chronic inflammatory condition. And with that, I just wanted to finish by highlighting this a little bit more recent work, where clearly highlighting again that mucus-associated bacteria are very important to drive the detrimental effect of dietary emulsifiers. Here what we did was, okay, we know that there is microbiota that are more or less resistant, so can we compare and can we switch a microbiota that's fully sensitive to a resistant status? And for this, we use the probiotic bacteria Akkermansia mucinifera, which is able to favor the mucus layer and to increase mucus production by the intestine. And very briefly, because I'm running out of time, what we were able to report is by giving this type of probiotic to mice that are normally sensitive to dietary emulsifiers, you can fully convert them to being fully resistant to dietary emulsifiers. So again, highlighting that this mucus-associated bacteria, this mucus-associated microbial population is very central in driving emulsifier sensitivity and downstream chronic inflammatory condition. And with that, I'm going to script. I mean, this is really our model, that we are not all equal when facing this type of compound, and that, again, the mucus-associated bacteria is very key to drive sensitivity or resistant status. I wanted to thank all the people involved in this work, and especially the DIM for supporting this cell sorter that will allow us to isolate and identify those mucus-associated bacteria and to which extent they can drive chronic inflammatory condition. And with that, I'm done. Thank you for your attention, and I think we'll have time for a few questions. Thank you.
Any quick question to Benoit? So I'll ask a very quick question. What's your model? Is it the genetics of the host that sort of selects whether they have resistant or susceptible bacteria, or it's their diet?
Yeah, that's a very good point. And so far we have no idea. We just know that all the microbiota are not going to respond the same. What is the driver of that? It can be genetic. We think it's going to be mediated by the diet. So we think that our past history of exposure to those food additives will shape a more or less sensitive microbiota. And for this, we are currently collaborating with the Nutrinet Santé cohort, where we analyze the microbiota from 10,000 participants, on which we will know exactly their status in terms of exposition to those food additives, and we will be detecting in the lab if they're sensitive or resistant. So we are working on this aspect. Yeah.
Thank you very much.
Thank you.
And